Hi guys, I'm back with you here to do the final video in chapter 12, and sorry about that. Do the final video in chapter 12, and this final video deals with the regulation of the cell cycle through molecular control systems. It turns out that how often cells divide can be different based on which type of cell we're talking about or which type of cell is under study. And the differences actually result from regulation at the molecular level. So based on whether or not it's a heart cell versus a liver cell or whatever, those different cells are going to want to be, the cell cycles in those cells are going to be different based on where they're at, and they need to be regulated as such. So in this final section of the notes, we're going to talk about a little bit how those signals are actually regulated. Okay, so there is significant evidence for the fact that there are cytoplasmic signals that take care of this. It's very similar, it's kind of connected to what we talked about in chapter 11. And so I wanted to walk you through an experiment that kind of outlines or shows this this evidence for regulation of the cell cycle using chemical signals that are present in the cytoplasm. Okay, and it deals with fusing mammalian cells with each other, but they happen to be in different stages of the cell cycle. Okay, and so we're going to look at experiment 1 here. It's kind of like the control. And if you look at experiment 1 here, we're going to take this cell right here that's kind of background in blue, and it says that this cell is in the S phase. And so we know that this cell right now is actually replicating its DNA. And we're going to fuse it with this cell that is in the G1 phase. Okay, so it's the G1 phase is one phase prior to the synthesis phase, right? And so we're going to see what happens, and we're going to fuse the two plasma membranes, and basically what that's going to do is going to cause them to share their cytoplasm. Okay, and so let me bring up a copy of this so that I can write on it a little bit. And what you'll notice is that when that's kind of cool. All right, what you'll notice here is that when we fuse these cell membranes, okay, ultimately these membranes will fuse, that this cell that was in G1 phase all of a sudden transforms to a cell that was in S phase. Okay, and so what they hypothesize is that whatever was in this cell, okay, some kind of chemical in this cell that denoted by the red dots there, very well may have transferred over to the other side, the other cell. Now, inherently there's something a little bit wrong with this. I'll see if you can't guess it. I'll pause the video and think to yourself, okay, well, is there something else that could cause this cell that's in G1 to go to the S phase? Okay. The answer is yes. There is something else that could cause it to go to the S phase. The answer is that it's in G1 and the natural next phase is the S phase. So maybe they just happened to get this cell that was in G1 very close to when it was going to go to S phase anyway. And so maybe it just transitioned at the exact moment that it was fused with the other one. Who knows? But there is evidence, and they go through a second experiment that kind of solidifies this in our mind, because it goes around the issue that we have with this experiment here. Okay, so they take a cell that is in the mitotic phase, M phase. Okay, we can see that because it's actively condensing its chromosomes. The centrosomes have created microtubules and they're moving. Okay, so they're in the active dividing phase. We're going to fuse it with the same cell, G1. We're going to go to the G1 phase. We're going to fuse the cell membranes together. We're going to see what happens. And it turns out that this cell that was in the G1 phase all of a sudden flips and turns to a cell that is in the mitotic phase. And so this kind of gives them some solidifying evidence that says, okay, there's some kind of thing, some kind of chemical signal that's in this cell that once we fused their cell membranes, it went and moved over to this side, and it made this cell that was in G1 turn into a cell that was in the mitotic phase, okay? And so this gets around that issue of the previous example, the previous experiment, where maybe we just got a cell that was in G1 and it was going to go to the S phase. But in this case, it totally skips the S phase, skips the G2 phase, and goes right into mitotic phase, okay? And we know that it skipped the S phase. Now I'll give you a second, pause the video, see if you can figure out how this cell right here skipped the S phase. See if you can figure that out. It turns out that this cell actually only has one copy of the DNA. If we look in there, it's only got one chromosome or one sister chromatid, whereas this one over here has two sister chromatids, indicating that this DNA has replicated, whereas this DNA has not. Okay, so this cell right here totally skipped the S phase. It did not even replicate its DNA. So there is evidence for these chemical signals that drive the cell cycle. Okay, because we looked at two of them here, one in the S phase, one in the M phase, that allow these cells to get proper messages to do proper things during those phases. Okay, so kind of a really neat experiment when you're talking about evidence to support this idea of cell signals dictating when a cell goes into a certain phase or not. Okay, so the cell cycle control system is a sequential set of events, and they are directed by the cell cycle control system. It's very similar to a clock with gates. And so a clock with gates would look like, okay, if you got to a certain time, it checks to see the clock's still working, checks to see the time is still good, hits a gate, and then once everything is good to go, the gate falls down and the clock can keep moving. That's kind of what this is going to look like as we go through. So this clock has specific checkpoints where the cell cycle kind of stops, gathers itself, says, okay, is everything good to go? We've got the proper amount of stuff. Are we big enough? Have we replicated our DNA? And if there's a go-ahead signal, then it will continue its process. Okay, so in your head, when you're thinking about the cell cycle, think about it in terms of something that looks like this, that as this thing progresses, maybe there's this almost like a timer switch in the middle. As this thing progresses through time, it's going to go through all these different events, and it's going to say, okay, we went through all this stuff. Let's hit this gate. Let's see what we've got. Okay, let's see if everything's good. Do we have enough DNA? Is the DNA starting to condense? Do we have enough organelles? Do we have enough cytoplasm? Okay, we've got all that. This gate will then fall over, and then this little timer will be able to progress through mitosis. Okay, so we would start this timer. I should have started at the beginning. We'd start this timer at the very beginning of the G1 phase, and it would have gone moved all the way here. Okay, are we big enough at the G1 checkpoint? Are we big enough? Are we starting to make progress on creating proteins and making organelles and getting ready for cell replication? And the cell says yep, and then this gate falls over, and then the timer can progress. It'll go through synthesis, and then it'll go through G2, and then it'll hit another checkpoint gate here to make sure everything's good to go. All right. So the cell cycle control system seems, through experimentation, seems that as if the G1 checkpoint is reached, it seems as if the rest of the checkpoints will kind of go ahead as if nothing's wrong. Okay, so apparently there's a lot of events that happen in G1, the first gap phase, that are really important to synthesis and really important to G2 and also mitosis. And if all of these things happen in the G1 checkpoint, then usually everything else will go off without a hitch. Now, if a cell doesn't receive this go-ahead signal at one of the checkpoints, that cell will enter a non-dividing state called the G0 phase. Okay, and the G0 phase, a G with a little 0 at the bottom, is one we haven't discussed yet. And the G0 phase basically is this non-dividing state. It's a state in which the cell can kind of pause, take a little bit of a break, try to figure out what's going on, what has happened, why are things incorrect, why are things wrong, are we missing something, and then if it gets the correct signal and things get fixed, it can then re-enter the cell cycle and continue on its way. So the G0 phase is really more than anything is a state of the cell where it can just take a breather and figure out what went wrong, try to fix it, and if it can fix it, then it'll go back to doing what it was doing. Okay. Now there are some cells that are perpetually in G0 phase in our bodies. Once we reach a certain maturity level, once we reach a certain age, or our body has matured to a certain point, our nerve cells and our muscle cells, generally speaking, now there are obviously some exceptions to this, never divide again. Okay, they never regenerate. Okay, I know there has been some studies done where they are kind of getting out of this idea that nerve cells, for instance, are perpetually in the G0 phase. Some nerve cells they've noticed in rat brains actually regenerate, and you would imagine if rat brains can do it, human brains probably can as well. So there is some evidence to state that mature nerve cells can maybe revert to a dividing state so that they can replicate again. As far as I know, there's not been any research done on muscle cells to say that they would then also start to divide again. And that's not to say that people you see with giant muscles, right? Big bodybuilder, steroid freak guys or girls are producing more muscle cells. They still don't produce muscle cells. What they actually do is they produce more proteins inside of the muscle cells, and that's what builds the muscles even bigger. Okay, so it's not like they have all of a sudden taken some drug that causes them to have more muscle cells. They just break down the proteins and rebuild the proteins in their existing muscle cells to make them bigger. Okay, so that's maybe a common misconception. But regardless, these checkpoints, if they're not met, the state enters this non-dividing state, and as we can kind of see in this diagram here, this non-dividing state kind of puts it on hold. It says, okay, let's see if we can figure this out, and then once we figure it out, it can then lower this gate and go on with the rest of its cell cycle. One of the issues that you might see take place, for instance, is a kinetochore microtubule. This was discussed in the last video. A kinetochore microtubule, maybe it doesn't get attached to one of the chromosomes, and there's no way for that chromosome to get back to where it has to get back to, once the anaphase hits and it starts to divide. And so that would be an example of something that happens during mitosis, during the phases of mitosis, that would cause some kind of checkpoint not to be met, and maybe it sends another microtubule out there and attaches it, and then everything can go off and be normal as if it had attached in the first place. So it gives it some time to figure out. More often than not, if it enters this G0 phase, if it can't figure it out, if it can't figure this situation out, it will kill itself. Okay, because there's nothing worse than a cell that goes through cell division and then you've got this mistake. You've got a daughter cell that has too much DNA or a daughter cell that has too little, and we know that we'll find out here that can cause some issues. Okay, you're missing genes in a cell can cause some issues. All right. Now we're going to talk about some of these cycle signals. One of the first ones we're going to talk about in depth is listed in front of you, and then we're going to talk about some other types of stop or go signals that are found in the cell that are a little bit less in detail. Okay, so the one that's in front of you is called the cyclin and cyclin-dependent kinase signaling pathway. All right, and this relies on two types of regulatory proteins, and they are known as cyclin is one, and then cyclin-dependent kinase. Okay, so these two things you can imagine just by their names kind of work together. Cyclin-dependent kinase depends on cyclin, right? So they kind of work together. Now the activity of these cyclins and the CDKs, cyclin-dependent kinases, fluctuate during the cell cycle. During different parts of the cell cycle, these things aren't needed, so they're broken down and their parts are reused for other things in the cell, while other parts of the cell cycle, these things are needed, and they are built up and combined. Okay, so when these things actually do work together, cyclin is going to combine with CDK. Okay, it makes sense. Cyclin-dependent kinase is going to come together with cyclin to make something known as the maturation promoting factor. Okay, so it's got this like this crazy set of things that all come together to make a bigger set of things afterwards, okay? And so this MPF is kind of the final molecule that's created once all these things come together. And it's the actual thing that does work during the cell cycle, okay? So the maturation promoting factor, once it comes together to make this molecule called the MPF there, it actually phosphorylates different proteins seen in the cell cycle so that certain actions actually occur. And we talked about in chapter 11 the importance of this phosphorylation, right? Phosphorylation, when you phosphorylate something, you add a phosphate from ATP to another molecule. And when you add that phosphate to this other molecule, it activates it, right? And so this activated molecule can then go do all sorts of cool stuff, all right? And so this MPF is going to phosphorylate different things that happen in the mitotic phases. It might phosphorylate the motor proteins that are found in the kinetochores. Remember, we talked about that in the last video. The motor proteins need ATP to work, to move the chromosomes from the center of the cell to one of the poles. Okay, so that might be an example of where MPF comes in, phosphorylates a motor protein, and then that motor protein can move that chromosome back towards the pole of the cell. All right, so how does this look, and where do these things start to accumulate, and when do they come together in the cell cycle? That's what we're going to talk about here in this picture. And let me see if I can get a drawing up of this so I can draw all over it. All right, that's not ideal down there, but oh well. Actually, let's do it proper. Look at that. That's a stupid look, isn't it? Anyway, this diagram shows us how these things called cyclins and CDKs fluctuate during different phases of the cell cycle. So they've got all this stuff overlaid. I'm going to spend more time here on this bottom diagram that you see. They have these different molecules overlaid with the cell cycle through time. Okay, so let's start here when we've just made a brand new cell. At this point, CDK right here and cyclin, this dark purple stuff, are no longer needed. We just got done with cell division. We don't need to do anything else in mitosis because we just got done with mitosis. So the CDK, which we see right here, breaks off of the cyclin. And then the cyclin's actually degraded. It's broken down. It's not needed at this point. So the cell can actually use these parts that we see right here for other things until they are needed. Okay? So the cell goes about its business. We don't see anything happening here. There's nothing built up. There's no molecules of any import that have started to be created. We go through the synthesis phase. We make some DNA. And okay, we're done with the synthesis phase. Now we start to see in the G2 phase that CDK comes back into the picture. Okay? And so does cyclin. These two things start to accumulate at a higher rate during the beginning to middle of the G2 phase, okay? Because the cell notices things are starting to happen. We've made some extra DNA. We're going to have to start to put together this CDK and cyclin because we have to get ready for mitosis. We're going to start to divide here soon. So by the time we get to the middle of the G2 phase here, okay, it kind of lines up with this point of the arrow where CDK and cyclin come together to make the MPF. Okay? The maturation promoting factor is this kind of like Voltron Power Ranger molecule. It's got the cyclin that's come together with the CDK to make this bigger thing called the MPF, which can actually do stuff. Okay? So at this point, right at the end of the G2 phase, we see that we've got maturation promoting factor has been built, and that's good because maturation promoting factor is going to be utilized during the mitotic phase, which we see here. And again, the mitotic phase has us using the MPF to phosphorylate these different activities during mitosis. Maybe, like I said before with the previous slide, maybe it's we are phosphorylating the motor proteins to get those chromosomes back to the poles. Maybe we're phosphorylating other motor proteins that cause the movement of other things throughout the cell. Okay, so this maturation promoting factor is important to basically fueling the events that happen in the mitotic phase. Okay? Now, once mitosis gets done, once we've made our two daughter cells, this maturation promoting factor is no longer needed. It's done everything that it needs to do, so we're going to break it apart. We're going to break up the band, get rid of them. Okay? Cyclin is going to be degraded. You see the cracks in the armor there, and it breaks apart. And then CDK goes about its business and does something else until it's needed again in the G2 phase. So I've drawn all over this stupid diagram here in an effort to get you to understand that during certain phases of the cell cycle, these molecules, cyclin and CDK, are not needed, whereas other phases of the cell cycle, they are needed and they start to accumulate and assemble themselves. Okay? And we can see this same assembly of cyclin and CDK into the MPF. We can see this same fluctuation through different cycles. Here's one cycle of the cell cycle. Here's another cycle of the cell cycle. And as the cell goes through these waves, you'll notice that cyclin increases and then it decreases because it's no longer needed. MPF increases during mitosis and then decreases because it's no longer needed. Okay? And so these very important regulatory molecules break apart when they're not needed, and they reform and are used when they are needed, and they help to make events occur during mitosis. Okay, so that is how that process works. We do see other stop and go signals, these internal and external checkpoints during the cell cycle. So I just kind of want to go through these just very generally. They're nothing super in depth like we just went through. But I want to make sure that you understand that there are other ones there, and I'm sure there's dozens of other signals that have to happen before you actually have a properly divided cell. Okay, so an example here of an internal signal is when the kinetochores, and we talked about those, don't actually attach to the spindle microtubules, and they send a molecular signal that says, hey, stop, we're not attached to any microtubules, we need you to halt the movement of anaphase, somebody come get us. And so the cell will send out some more microtubules, and eventually those microtubules will attach to the kinetochores of the chromosomes so that those chromosomes can move their way back to the cell's pole. Okay. Some external signals are growth factors. Okay, and these growth factors come from, like I said, the exterior, the environment. And they are proteins that are released by some cells that stimulate the division of other cells. Okay, so growth factors are examples of external signals that can help drive the cell cycle. Okay, and so they will tell cells to, okay, you're not currently dividing. I'm sending a message from my buddy over here. He says that you need to come in and divide like the rest of us. Okay, so some of these external growth factors are important as well. A very specific example of this is called PDGF. Okay, platelet-derived growth factor. You don't need to remember what PDGF stands for other than the fact that it's a growth factor. Okay, and this stimulates the division of fibroblast cells in human cultures. Okay, this diagram outlines how PDGF works. I'm going to let you read it. I'm not going to spend a whole lot of time analyzing it. If this is something that you would like me to explain to you in class, please by all means let me know. That's what class time is for. That's why we do these lectures kind of a little bit quicker here, and class time is for discussion of diagrams like this. But PDGF, once administered to cells, stimulates them to divide. Okay. Another example of an external signal is density-dependent inhibition signals, and this tells crowded cells to stop dividing. We're going to cover this one more in depth as we work our way through the next slide, okay? And also we have some animal cells that also exhibit something called anchorage dependence, which tells cells that if you're not attached to something, you don't need to be dividing right now, okay? Now, cell anchorage and density dependency are things that cancer cells do not take heed of, okay? And part of what we're going to talk about here is cancer and how cancer actually gets going because of these mistakes in cell cycle control, okay? So this one I will probably go ahead and describe to you a little bit here, okay? Now, normally in cells, cells in culture, and really cells in our body, cells in our body need to be anchored to something before they'll actually start dividing, okay? And so that's what we see up here. These cells at the top are anchored to the dish surface, okay? And once they're anchored to that dish surface, they will start to divide and they'll fill the layer of the dish, okay? Now this is where density dependence is exhibited, okay? These cells that have basically filled the entire plate are going to send signals to the other cells that are surrounding them that says, okay, we're really close to each other. We no longer need to divide because we've filled the entire plate. That's effectively what that cell signal says when they have this density-dependent inhibition. And so that cell signal is perceived by all the other cells as an indicator that says, stop reproducing, stop dividing. Okay, so that's the cell response. Now, if some of those cells are removed, like we see here, to kind of mimic maybe tissue damage, like you've cut yourself or something like that, this guy is removing some of these cells here, leaves a hole in this tissue layer. The cells around it are going to send out a signal that says, okay, there's a gap in our tissue. It sends a signal that says, okay, we need to start dividing to fill in the gap. And so then it fills in the gap, and then it says, okay, we're done, we filled the gap. Send out that density-dependent inhibition signal for everybody to stop, okay? That's how normal cells exhibit density dependence or anchorage dependence. They have to be attached to something to divide, and then they have to be comfortable with the amount of cells that are there, okay? And cancer cells don't exhibit any regard for the rules in as it pertains to those two particular types of inhibition, those two particular types of signaling. Cancer cells, and this is where you get into things like blood cancers and metastatic cancers and things of that nature, blood cancers create cell division just floating through your bloodstream. They don't have to be anchored to anything, okay? The same thing with some of these metastatic cancers. They can actually pick up shop, move from where they started, and divide on their way to the next place where they're going to set up shop, okay? And so some cancers don't even need to be anchored to a tissue. They'll just divide wherever, okay? And then on top of that, cancer cells don't exhibit density dependence, right? And this makes a lot of sense because we see many people who have cancer have tumors, right? And tumors are nothing more than an abnormally large amount of cells that are there because there's just uncontrolled cell division. Okay, so something in the cell cycle control system is broken that causes them to just continuously divide and stack up on top of each other. And that's what we see down here in this diagram at the very bottom. We see just a giant chunk of cells that shouldn't be there. Normally in this dish, the cells are going to fill up just the bottom layer and then stop dividing. And if this is a cancerous cell or a cell with a broken cell cycle control system which does not exhibit density dependence, these things are just going to continue to divide over and over and over again. They're going to stack up on top of each other and pile up on top of each other, and that can be really, really dangerous, especially if you get it in, a really important organ system in your body, your kidney, your liver, your lungs, obviously, your brain, those places. Obviously, those types of tumors can be devastating and deadly, right? And so it's because of broken cell cycle control systems that we have cancerous tissue, okay? Now, excuse me. Cancer cells don't normally respond to these control mechanisms, and as a result, they form tumors. And they can pretty much form these tumors anywhere because they don't need to be anchored to anything. And they'll continue to divide unhindered until the organism is dead because of what they're doing. Now, if these abnormal cell masses remain in the original site, okay, that tumor is called a benign tumor. Now, generally speaking, if you get a tumor that is benign, you got one that is okay. I don't like saying okay because benign tumors just mean that the tumor is going to stay in the original site. It doesn't mean that benign tumors aren't dangerous. You can have a benign tumor in your brain and it can still kill you. All right? But what benign means is that it's not going to move. Okay? And so usually benign tumors they can operate on. They can go in there and take it out, and usually people who get those removed, aside from maybe a big scar or maybe some damage that was caused beforehand, are usually okay as long as it's not in a particularly important organ system, okay? The ones that get particularly nasty are the malignant tumors, and those are the tumors that invade surrounding tissues and they metastasize, okay? Metastasizing means that that cancer has moved from one part of the body to the next, okay, where it oftentimes forms secondary tumors. Personal experience, my stepfather passed away of pancreatic cancer a few years ago, and the cancer started in the pancreas, but it moved basically everywhere. It moved to his lungs, it moved to his liver, it moved to his brain, everywhere. It was by the time he had passed away, it was everywhere. And so that's people who have loved ones who have passed away or dealt with this know how scary malignant tumors can be. And it's why when you hear a doctor say your tumor's benign, you're like, breathe a sigh of relief, because you know that tumor is not going to be transferred to another part of your body. It's at least going to stay put. And that's where the danger from cancer comes in, is the fact that these things don't follow the normal rules. They're going to pick up shop and move up wherever they want. They're going to divide while they're moving, and then they're going to make these massive tumors when they get there. Okay, and so that's one of the reasons we see breast cancer so heavily, thought about, is because it is deadly, because it is a very quickly metastasizing tumor. Okay. If you catch breast cancer early enough in its development, you can operate on it. You can take drugs to get it. And sometimes people have their entire breast tissue removed. But for the most part, why it's so scary is because it moves very, very quickly. It'll start in the breast tissue, but then it moves to other organs of the body. And that's what makes it so dangerous and scary, because it starts in something like a tumor in the breast is not going to kill you, right? That is fine. If it was a benign tumor in the breast, they'd be like, okay, fine, whatever. But if it's one of these ones that likes to move around like these do, it needs to be dealt with very, very quickly. And that's why there's the advocacy for, in October of Breast Cancer Awareness Month to make sure people get checkups, right? To regularly check up on the breast tissue. I mean, I kind of say people on purpose, because we actually we have found out that men can get breast cancer as well. Okay, so everybody kind of gets it, but obviously women get it at a higher rate. Now, what we see here in this diagram is something especially sinister, which helps with the metastasization of this particular type of cancer, and really with all cancers. And it kind of goes to speak to the cell signaling and the cell communication as well. These cancerous tumor cells are really, really good at spreading, and what they do is they actually send out chemical messages through to the blood system, okay, to the blood and the lymph system. This is kind of like the highway of our body, right? And these chemical messages will cause the circulatory system, like right here, to actually grow towards the tumor. So it's like saying, hey, I'm kind of lazy, says the tumor, I want the roadway to be built towards me. And so it actually, once these circulatory vessels are then built, it allows this tumor to spread much, much easier as the cells hitch a ride into the vessels and then move to different parts of the body because of it. So that's how these things get around. They move by way of our lymph system and our blood system. Okay, both of those are circulatory systems that go all over the body. Okay, so cancer is a side effect of broken cell cycle control systems, and so you have to have a healthy respect for the fact that most of us have pretty robust, well-working cell cycle control systems. Pretty neat thing. Okay, so I think that's it actually for chapter 12. Sorry to end it on such a grim note, but that's why scientists do what they do and doctors do what they do, and that's why you guys learn what you learn, because a lot of you guys want to help people with this stuff. So I will let you go with that, and have a good afternoon. See you guys.